Hello and welcome to our video on section 17.4 where we're going to talk about pathogen recognition and phagocytosis. If you remember, phagocytes are going to be the cells whose main function is to seek, ingest, and kill pathogens through the process of phagocytosis. Now within this section, our learning objectives are to explain how leukocytes migrate from our peripheral blood into infected tissues, and explain the mechanisms by which leukocytes recognize pathogens, and explain the process of phagocytosis and the mechanisms by which the phagocytes destroy and degrade pathogens. So we'll start with the process of extravasation, which is also known as diapedesis. And this is going to be initiated when complement factor C5a is released by resident macrophages within the tissue that has been infected. So we can kind of see this right in here. We see our resident macrophage is releasing cytokines and also that C5a complement protein. And that is going to cause our leukocytes to stick slightly to the adhesion molecules that aren't really depicted over here, but we've got some adhesion molecules here that are almost like speed bumps that will slow down these white blood cells as they're rolling near this infected area. Now, the whole process that we want to take place is to slow them down, and then they will start flattening out and squeezing through cellular junctions. So what we see pictured here is a capillary. Now we can't have this happen in arteries or veins because we also have multiple layers here, like the tunica media and the tunica externa that has connective tissue and muscular tissue. So this is in a capillary, and it squeezes through between the endothelial cells so these white blood cells can move into the area of infection. And that point where they're able to pass through is called diapedesis. So now our neutrophils are arriving to the area within hours, and then within a few days we'll have some monocytes come in and become macrophages in order to go through the process of phagocytosis to kill off the bacteria that has infected here. Now let's talk about the process of pathogen recognition. So we've got to define some terms here. First, we have our PAMPs, which stands for pathogen-associated molecular patterns. So essentially, this is a molecular structure that our phagocytes can recognize that are common within many groups of pathogenic microbes. So here are some common ones that I've listed here. We know within bacteria, we have that peptidoglycan within their cell wall. We also have flagellin, which is a protein in bacterial flagella. LPS, we know, could be found in our gram-negative bacteria, and our lipopeptides, which are found in most bacteria, and if we have a virus, nucleic acids such as viral DNA and RNA stand out to our phagocytes. So these are all signs that we need to attack. Now, the other term we need to know is PRRs, pattern recognition receptors. These are structures that are found on our phagocytes. So imagine this is our phagocyte over here, and these green structures here are the receptors. So they are going to help recognize the PAMPs. So whether it's a lipopeptide, a flagellin, a peptidoglycan piece, right? And there are specific receptors called toll-like receptors, and these are located on the surface of the phagocyte. And I want you to also note that it doesn't have to be one of the PAMPs that we've discussed here that it can attach to. It can also be maybe from opsonization of a pathogen from an antibody, right? Putting that red X on a microbe saying, hey, you need to come kill this microbe. It could also be complement factors such as C1q, C3b, C4b, and lectins that assist our phagocytes in recognizing the pathogens and attaching to initiate phagocytosis. And also notice that we have this nucleic acid receptor, so it doesn't necessarily have to be something on the surface of the phagocyte, but it can also have receptors on the inside of the cell. But essentially, no matter where the reaction between the PAMPs and the PRRs take place, we are going to activate the phagocyte, which can then proliferate, essentially multiplying itself, and produce cytokines and enhance intracellular killing. Now let's talk about pathogen degradation, essentially what the process of phagocytosis is. But of course, first we needed to recognize that pathogen, and the pathogen becomes engulfed in a vesicle here. And this essentially is starting that process of phagocytosis. So this then becomes a phagosome, which is when we have the pseudopod, so almost like little feet coming and wrapping around the pathogen and pinching it into this vesicle, is when it becomes a phagosome. And it's going to fuse together with a lysozyme. So now when we've got both phagosome and lysozyme fused together, we're going to call that a phagolysosome. And that is going to allow lysosomal enzymes such as lysozyme, phospholipidase, and proteases to start digesting the pathogen, the bacterium here. There are some other enzymes that are going to help with the respiratory burst, and we can increase our uptake of oxygen and have production of things like superoxide anion, hydrogen peroxide, hydroxyl radicals, and other reactive oxygen species that are all antibacterial. So it goes through the process of degradation, and once it's complete, we've got some waste products that are left over, and it could be secreted out of the phagocyte. Now, we're not going to get rid of everything here, because another important function that these phagocytes have is that they can be antigen presenting cells. Specifically, our macrophages and dendritic cells are going to be these APCs. And what they do is they take a piece of the pathogen, like a antigen from a pathogen, and we'll talk more about what an antigen is in the next chapter, but essentially it's kind of like an ID showing that there's a microbe. It's like a wanted sign that the macrophage is going to take and display on the outside of its cell so that it can trigger our immune system further.